Good evening. Let's imagine the future where spinal cord can be easily repaired, and when Parkinson's disease and chronic pain can be treated without drugs or injury. Today, it may sound to you like science fiction, but I believe that those challenges in neuroscience can be overcome by inventions in material science. But let's just start from the beginning. What do Parkinson's disease, paralysis, and chronic pain have in common? All of these conditions involve faulty operation of the nervous system. To treat them, we must learn to communicate with its building blocks, neurons. So how do we do this today? On the screen, you see a picture of a deep brain stimulation implant. This is a clinically approved treatment for Parkinson's disease. For this procedure, a patient's head gets placed into a stainless steel stereotactic frame, and 2 holes are drilled through their skull. Through those holes, a physician inserts 2 2-millimeter thick, 8-centimeter long wires. These wires then apply voltages 1,000 times greater than are used by neurons for communication. And then the physician asks the patient, "How are you doing?" and checks for their tremor, because the patient is entirely awake during this whole procedure. And if the stimulation doesn't return appropriate results, the doctor will wiggle those wires and try again. And surprisingly, this therapy works. It's incredibly effective to restore movement to Parkinson's patients. The question is, at what cost? Because the side effects can be quite profound. A quiet, workaholic personality can be turned into an unstoppable, compulsive gambler. This, of course, should not be surprising at all, because the wires that are used in the deep brain stimulation implant are 1,000 times larger than neurons that they're trying to stimulate. But then this is, of course, a problem that modern electronics should be able to solve quite easily. In the end, your smartphone packs 100 million transistors into a square millimeter. So how come those transistors are not helping us to communicate with neurons and treat Parkinson's? Well, it turns out that communication between transistors in an integrated circuit and between neurons in the nervous system is fundamentally different. A transistor in an integrated circuit is connected to 3 other transistors, and it uses electrical signals to transfer and receive information. A neuron in the nervous system is connected to thousands of other neurons and uses not only electrical, but chemical, mechanical, thermal, and many other types of stimulus to receive and send information. In addition, integrated circuits are built on flat, rigid semiconductor wafers, while neurons are built of organic matter and form a soft, squishy, 3-dimensional mesh. So in order for us to learn to communicate with neurons, we need a device that can speak all neuronal languages and at the same time be soft and squishy, just like the neural tissue, and have the resolution of individual neurons. So how do we build this soft, squishy Swiss Army knife for neuroscience? Well, the inspiration comes from nerves themselves. They're flexible, stretchable, and fibrous. So in our group, to communicate with neurons, we have designed fibers. To build a flexible, multifunctional fiber, we start in the machine shop, and we design a model that is a few centimeters large in diameter, few centimeters long. We then take this model, we heat it, and we pull it. And the result is hundreds of meters of flexible, multifunctional fibers with microscale and even nanoscale features. In fact, our fibers are as thin as your hair. Pick one up. And we can wrap them around the tip of an individual pencil, but inside they contain conductive electrodes for electrical communication with neurons. They contain optical waveguides for optical communication with neurons, and they contain microfluidics for chemical communication with neurons. We can even deliver genes. What you see on the screen is a recording from a brain following a delivery of a gene that slowly makes neurons sensitive to blue light. As you can see, not much is happening yet. But let's wait a few days. Specifically, let's wait 11 days. And you see how the activity in response to blue light pulses begins to emerge. And it becomes more and more robust as the time goes on. In this experiment, a hair-thin fiber that has delivered a gene and delivered the optical pulses has also recorded neural activity that you see on the screen. We can make these devices not only of flexible polymers, but also of stretchable devices, make them be able to withstand extreme deformations and conform to arbitrary shapes. And you can see how this device can be implanted in the spinal cord and withstand the stresses that your spinal cord experiences, not only when you take a step, but when you stretch out in a yoga class. But let's just not get carried away here. These devices, no matter how capable, multifunctional, miniature, and flexible they are, we still require a surgery to implant them. What if it was possible to communicate with neurons without implanted hardware? What if we could do it wirelessly and entirely non-invasively? To do this, we must learn from neurons again. The way neurons perceive different stimuli is through their ion channels. Those are proteins on their membranes. When an ion channel receives a stimulus, it opens to ion flow from outside of the neuron and from the neuron outside. And through this process, neurons can become more active or less active. So if we want to control neural activity, we must learn to speak the languages of those ion channels. And one of those languages is heat. So how does that work? Well, imagine eating a chili pepper. The burning sensation on your tongue is exactly the same sensation that you experience from a boiling cup of tea. And the reason for that is the ion channel that responds to capsaicin, which is the spice in chili peppers, is exactly the same ion channel that perceives heat. And it turns out this ion channel is present not only in your tongue, in the receptors in your tongue, but also in neurons in your brain. So what I'm saying is that if we want to control neurons, all we need to do is to heat them up. Of course, we don't want to heat up the entire brain or the entire spinal cord, for example. So this means we need a signal that can penetrate deep into the body without interacting with our tissues and without being attenuated. And then we need a transducer that can convert that signal into heat locally next to neurons. A convenient signal is a weak magnetic field, 1,000 times weaker than a magnetic field in a magnetic resonance imaging machine, an MRI machine. So now, to convert that weak magnetic field into heat, my team developed magnetic nanoparticles. Those are tiny specks of iron oxide, which is essentially a magnetic form of rust. They're 1/5,000th of your hair in diameter, and they're similar to MRI contrast agents. And we can dissolve them in water, just like a drug. But when we place them into a weak magnetic field that alternates at a very high frequency, they undergo a process called hysteresis, which is essentially a magnetic analog of friction. And through this process, they dissipate heat. So now, if we want to stimulate neurons arbitrarily deep in the brain, all we need to do is to deliver those particles to a neural circuit of interest and then apply magnetic field to the entire subject remotely and wirelessly. What you see on the screen is a movie of neurons firing or being active in response to magnetic field in the presence of nanoparticles. In this experiment, there are no wires, no connectors, and no holes in the skull. So what is next for us? The advances in neural interfaces that I have shared with you this evening were inspired by biology, but were enabled by materials discovery. And I believe that if we continue learning from the complexity of the nervous system, and if we continue inventing new materials, we can not only advance our basic understanding of the neural circuits, but get closer to a future when Parkinson's disease, paralysis, and chronic pain are simply a thing of the past. Thank you.